Welcome to this lecture on carbohydrate metabolism. Today, we are going to explore the magic of glycolysis. This is the fundamental process that keeps almost every living cell alive, including your own. Whether you are running a marathon or just thinking hard, your cells are constantly working. Let us dive in and see exactly how our bodies extract energy from the food we eat to power our daily lives. To understand why this process matters, imagine you have a massive, roaring bonfire, and you want to use its heat to boil a tiny cup of water. If you throw the cup directly into the fire, the water instantly vaporizes, and the energy is completely wasted. In your body, a molecule of sugar called glucose is exactly like that roaring bonfire. It is packed with a massive amount of chemical energy. If your cells released all that energy at once, you would literally overheat, and the cell would be destroyed. That is why carbohydrate metabolism, specifically glycolysis, is so brilliant. It breaks down that sugar step by step to capture the energy safely. Now that we understand why we need to break down energy slowly, let us look at what glycolysis actually is. Formally, glycolysis is a 10-step metabolic pathway that takes place in the cytoplasm of almost every living cell. The word itself comes from the Greek roots glyco, meaning sugar, and lysis, meaning to split or break apart. During this carefully orchestrated process, a single 6-carbon molecule of glucose is systematically split into two 3-carbon molecules known as pyruvate. Along the way, the cell actually has to invest a little bit of its own energy to get the chemical reaction started. However, it ultimately harvests a much greater amount of energy by the end of the pathway. This step-by-step breakdown leads us to an important question. Why is this specific pathway so incredibly important to your survival? First and foremost, glycolysis is the absolute foundation of all cellular energy production. It acts as the universal first step, whether you are a human, a plant, or a microscopic bacterium. It is uniquely powerful because it does not require oxygen to function. This means your muscles can still generate energy during intense, breathless exercise, like sprinting. Furthermore, certain vital cells in your body, such as your red blood cells and the neurons in your brain, rely almost exclusively on glucose and glycolysis to stay alive. Without it, complex life would be entirely impossible. Before we move on to calculating energy, let us look at the net chemical equation. This equation summarizes the entire 10-step process into a single mathematical representation. Conceptually, it shows us exactly how the raw materials are transformed into the final products. On the left, we start with glucose, our primary 6-carbon fuel source. On the right, we see it has been split into two molecules of pyruvate, which are 3-carbon molecules. We also see protons and water molecules released as natural byproducts. If the amount of starting glucose increases, every single product on the right side of the equation will increase proportionally. This balance allows scientists to track exactly where every single atom goes. Now that we have seen the main equation, let us break down the energy carriers involved in this process. Think of adenosine diphosphate, or ADP, as an empty battery. The inorganic phosphate is the physical piece needed to charge that battery. When they combine, they form ATP, which represents our fully charged energy batteries, ready to power the cell. Similarly, NAD+ is like an empty electron carrier taxi, just waiting to pick up high-energy electrons. Once it picks them up, it becomes NADH, representing the full electron taxis carrying energy to other parts of the cell. These molecules are the actual harvested energy that the cell gets to keep and use. Understanding these energy carriers leads us to a very practical formula. How do we calculate the total energy produced from a specific amount of food? Conceptually, this formula comes from the stoichiometric ratios in our net chemical equation. TATP represents the total ATP produced, and Ng represents the initial number of glucose molecules. We multiply by the constant 2 because the net yield of glycolysis is exactly 2 ATP per glucose molecule. The gross production is actually 4 molecules of ATP, but we must account for the initial cost of 2 ATP used in the investment phase. If you increase the number of glucose molecules, the total ATP produced scales up directly. Now that we have our formula, let us see this with a concrete example. Suppose you eat a small piece of candy and exactly 50 molecules of glucose enter a muscle cell. We are given that Ng equals 50, and we are asked to find the total net molecules of ATP produced. We choose our straightforward multiplication formula because the ratio of glucose to ATP is always 1 to 2 in this specific pathway. We substitute our given value of 50 into the equation and multiply by 2. This gives us a final answer of 100. Physically, this means the muscle cell has successfully charged 100 energy batteries to power its contractions, allowing you to move. Following our ATP calculation, we must perform the exact same calculation for our electron carriers to find the total NADH. We're still working with the 50 molecules of glucose given in the previous step. Because the net chemical equation shows that 1 glucose yields 2 NADH, we use the exact same multiplication approach. We multiply 50 by 2, giving us a final answer of 100. Physically, this means 100 electron taxis are now full. They will travel to the mitochondria to cash in their stored energy later, if oxygen is present. This demonstrates how a small amount of sugar translates into a massive amount of cellular activity. So we have seen how free glucose is processed. This brings us to a slightly different, highly practical scenario that happens in your muscles during a sprint. Instead of using free glucose from your blood, your muscle cell breaks down stored glycogen. When glycogen is broken down, it enters glycolysis already carrying a phosphate group. This is a massive advantage because it means the cell gets to skip the very first ATP investment step. By avoiding this initial cost, the overall energy profit for the cell increases. This reveals exactly why our bodies go through the trouble of building complex glycogen molecules instead of just storing free glucose. To quantify this glycogen advantage, we use the net energy profit formula. Conceptually, this is just like a business, where you subtract your expenses from your revenue to find your take-home profit. Y net is the net ATP profit the cell actually gets to keep. Y gross is the total enzymatic output of ATP generated at the end of the pathway. Finally, IATP is the initial ATP invested to get the reaction started. It is crucial to always distinguish between gross and net values in biochemistry. If we reduce our initial investment, our net profit naturally increases, which is exactly what happens when we use glycogen. Now that we have our profit formula, let us calculate the new net yield for glycogen. We are given that the gross production remains 4 ATP, so Y gross is 4. Because glycogen enters already phosphorylated, the cell only needs to invest 1 ATP instead of the usual 2, so our investment is 1. We subtract the single invested ATP from the 4 produced. This gives us a final answer of 3 ATP per glucose unit. Physically, this mathematical difference explains a profound biological phenomenon. By saving just 1 molecule of ATP per unit, the cell becomes significantly more efficient at generating energy. Let us apply this improved efficiency to our specific problem. We're asked to find the total net ATP yield if 10 units of glucose derived from glycogen enter glycolysis. We know our starting amount is 10, and we just calculated that each unit yields a net of 3 ATP. We multiply 10 by 3 to find the total energy. The final answer is 30 molecules of ATP. Physically, this means the muscle cell gains a massive cumulative advantage over time. This extra energy can literally be the difference between winning a race and collapsing from exhaustion. It proves that even the smallest changes in molecular investment yield dramatic physiological results. To visualize everything we have just calculated, let us look at this flowchart of the glycolysis pathway. Notice how the diagram is split into 2 distinct sections. At the top, in the red box, we have the investment phase. Here, you can see glucose entering, and arrows showing ATP being consumed to prime the molecule. Then, the pathway splits, leading into the green box at the bottom, which is the payoff phase. Here, you can see the arrows pointing outward, representing the production of ATP and NADH, finally ending with 2 molecules of pyruvate at the very bottom. This diagram perfectly illustrates the concept of spending a little energy to harvest a lot more. We have covered a lot of ground today, so let us summarize the most important ideas. First, carbohydrate metabolism begins with the universal and essential pathway of glycolysis, which safely extracts energy without needing oxygen. Second, remember the core transformation, carefully splitting a 6-carbon glucose molecule into 2 3-carbon pyruvate molecules. Third, we learned that this process yields a net of 2 ATP and 2 NADH molecules per free glucose, but it becomes even more efficient when using stored glycogen. Mastering this foundational concept is the absolute key to understanding how all living things power their existence. You now have a solid understanding of glycolysis.